In this video, I'm going to provide a mini tutorial on how neurotransmitter is released from vesicles at chemical synapses. Well, let's begin by looking at a cartoon. Here's my cartoon of a chemical synapse. We have the presynaptic cell shown on the left, postsynaptic cell on the right, and if an action potential arrives at the nerve terminal, then we'll get those vesicles to fuse and transmitter will be released. This process is going to be initiated when the action potential arrives at the nerve terminal. There are voltage-gated channels in the axon. Those will be opening up, sodium will come in, and then a wave of positivity can spread into the axon terminal itself where there are voltage-gated calcium channels. These will open up in response to the increased voltage. Calcium will enter the cell, and then calcium leads to a fusion of the vesicles so that we get release of the neurotransmitter. Those can then go bind to the receptors on the postsynaptic cell, and we'll get signaling. Now this process sounds simple, but it actually requires a fair amount of specialized machinery. So let's look at a blow-up of the terminal so I can show you that. Well, here's my cartoon showing a single synaptic vesicle at the terminal, and I'll just orient you by showing this is the cytoplasm, that's the synaptic bouton, out here's the synaptic cleft, here would be the plasma membrane, and then we've got our vesicle that's full of neurotransmitter. For these two membranes to fuse, the vesicular membrane and the plasma membrane to fuse, we need to have a whole bunch of specialized machinery, and collectively these are called snares. There are vesicular snares, those will be found in the vesicle membrane, and target membrane snares, or T snares, which in this case will be found in the plasma membrane. And I'll just draw some of them. So here are some of the vesicular snares. There's one called synaptobrevin. Here are examples of some target membrane snares. This is syntaxin and SNAP25, and then we're going to need another player here, synaptotagmin. That's going to be important for binding the calcium. All right, well, how are these going to act? Well, even before the action potential arrives at the terminal, there's the possibility to have these vesicles basically held ready to be released in what's called a docked and primed condition. For example, there's a protein called MUNC18, which can come and associate with one of the T snares, leading to a conformational change, and that then allows the T snares to associate, forming what's called a complex. Here the vesicle is docked at the membrane because of the association between the V snares and the T snares, but you can see that the two membranes haven't fused yet. This protein is docked, but it's not ready to be released. That process will require an influx of calcium. To do that, we need to have voltage-gated calcium channels. Now, when an action potential arrives, what's going to happen is calcium will enter through the voltage-gated calcium channel. Calcium can associate with a number of different proteins, including these calcium-binding domains on the synaptotagmin. And when the synaptotagmin binds the calcium, it undergoes a conformational change, and that allows that protein then to become associated with the other V and T snares, and this allows fusion of the vesicle membrane with the plasma membrane and release of the contents. Get exocytosis. Now the transmitter will diffuse across and it can interact with the postsynaptic cell, but in the meantime the presynaptic cell has to recycle this machinery so that it will be able to reload it. It does this with the help of even more proteins. So that proteins will come in, they'll associate with the bit of the membrane that was a vesicle, and then take that membrane and endocytose it to bring it back into the cell. And as this bit of membrane is taken away to be recycled and refilled with neurotransmitter, another synaptic vesicle can come to the active zone where it will be able to then fuse and release its contents. What happens next will depend on the particular receptors that are present in the postsynaptic cell. We'll leave that for a different video.